welcome back to the channel. In today's lecture, we are going to understand the basics of nucleotides and the nucleic acids. As you know, the amino acid sequence of every protein in a cell and the nucleotide sequence of every RNA is specified by a nucleotide sequence in cell's DNA. So let's begin by understanding what nucleotides are. Nucleotides are the organic molecules which are made up of three parts: a nitrogenous base, a pentose sugar, and a phosphate group. So nucleotides are three molecule compounds composed of the nitrogenous base, pentose sugar, and a phosphate. When a nucleotide lacks the phosphate group, it's called as nucleoside. So nucleoside is nitrogenous base plus sugar. Got it? In other words, we can also say that the nucleotides are nucleoside plus phosphate. So nucleoside plus phosphate makes the nucleotide. Okay, and the nucleotide lacking the phosphate group is called as the nucleoside. These nucleotides they serve as the building blocks of nucleic acids like the DNA and RNA. So DNA and RNA, which are the nucleic acids, they are made up of the long chains of nucleotides which store, transmit, and express the information of life. So these nucleic acids are also called as the molecules of life because they store, transmit, and express the information of life. Another important point here to remember is that these nucleotides not only act as the molecules that store and transmit the genetic information, they also have some other essential roles in the cell. For example, nucleotides, ATP, adenosine triphosphate, they serve as the energy carriers of the cell. So the nucleotides also act as the energy currencies of the cell. Okay. The other important role of nucleotides is that they act as the secondary messengers in the form of cyclic AMP or cyclic GMP. They act as secondary messengers in signal transduction. The other important role of nucleotides is that they act as the cofactors. Okay, for example, NAD, okay, NAD and FAD, which are the nucleotides, they act as the cofactors in the reductive biosynthesis of lipids. So now you have understood the role of nucleotides beyond their role in the genetic information or in the heredity. Got it? Now we will understand the composition of these nucleotides in detail. The first principal component of nucleotides are the nitrogenous bases. Okay, are the nitrogenous bases. So these nitrogenous bases are the organic molecules that contain nitrogen. Okay, that contain nitrogen atoms. Okay, so nitrogenous bases are the organic molecules containing the nitrogen atoms. Okay, and these nitrogen bases acts as the building blocks of nucleotides. Okay, building blocks of nucleotides. So you have understood that the nucleotides are the building blocks of nucleic acids, and the nitrogenous bases are one of the principal component or the building blocks of nucleotides. Got it? So what these nitrogenous bases are? They are organic molecules containing the nitrogen. It's of two types. Okay, these nitrogenous bases are of two types: purines and pyrimidines. What purines are? They are the double ring structures. So you can see here guanine and adenine, these are the double ring structures. Double ring, okay? 1, 2, 1. These are the double ring structures. And pyrimidine bases, for example, cytosine, thymine, and uracil, they are the single ring structure. So, to conclude that the nitrogenous bases are the nitrogen containing atoms, it is of two types, purines and pyrimidines. Purines are the heterocyclic double ring structures, whereas the pyrimidines are the single ring structures. Got it? So, the purine bases include the adenine and guanine. Adenine is also called as the 6-amino purine. 6-amino purine, okay, because it has a nitrogen, okay, it has a NH2 group here at the carbon number 6, okay, it contains nitrogen, okay, at the carbon number 6. So adenine is also called as 6-amino purine. The second example of the purine basis is guanine, okay. It's also known as the 2-amino, okay, 6-oxypurine, okay, 6-oxypurine, because it has the amino group at the carbon number 2. It has amino group at the carbon number 2 of the heterocyclic ring, and the keto group, okay, at the carbon number 6 here. Keto group is here, okay. So it is a keto group and the amino group. So the guanine is 2-amino 6-oxypurine because it has the amino group at carbon 2 and the keto group at the carbon number 6. The second type of nitrogenous bases in the DNA and RNA are the pyrimidines, which are the single ring structures, and the examples include the cytosine, thymine, and uracil. Cytosine, which is common in both the DNA and RNA, okay. Cytosine is present in both DNA and RNA. Cytosine is also called as 4-amino 2-oxypyrimidine, okay. 4-amino 2-oxypyrimidine, okay. Pyrimidine, okay. So thymine, which is present only in the DNA, okay, only in DNA. Thymine is present only in the DNA, and it's also called as 5-methyl 2,4-dioxypyrimidine, okay, 5-methyl 2,4-dioxypyrimidine, okay. When thymine lacks the methyl group at the carbon number 5, it's called as the uracil, okay. So uracil is 2,4-dioxypyrimidine, okay. Uracil is 2,4-dioxypyrimidine, okay. Got it. And uracil base is present only in the RNA, okay. Got it. So to conclude this is that adenine and guanine are present in both DNA and RNA, okay, and are double rings. Pyrimidines like cytosine, thymine, they are present in the cytosine is present in the DNA, and the thymine is also present in the DNA, whereas the uracil is present only in the RNA, and these all pyrimidine bases are the single ring system. So the important point to remember here is that the uracil is a pyrimidine base which is found only in the RNA, and the thymine is a pyrimidine base which is found only in the DNA, whereas the pyrimidine base cytosine is found in both DNA and RNA, okay. And the purine bases adenine and guanine, which are double rings, they are present in both DNA and RNA, okay. Also, the uracil lacks the methyl group at the carbon number 5. So you can remember that the uracil is the only, thymine is the only nitrogenous base which contains the methyl group, CH3 group. Okay. The second principal component of nucleotides is the sugar, okay, which is a pentose sugar. So pentose sugar is the second important component of the nucleotides. It's a 5 carbon simple sugar, that is C5H10O5. Okay. And also important point to remember here is that this sugar molecule connects the nitrogen base at the carbon number 1 and the phosphate group at the carbon number 5. So this pentose sugar acts as the base on which the nitrogenous bases and the phosphate groups are added. The nitrogenous base is added at the carbon number 1 of the pentose sugar, and the phosphate group is added at the C5 of the pentose sugar. Okay. Another important point to remember here is that these pentose sugars, okay, the pentose sugar can be present in the linear form, okay, or in the ring form. But in the nucleotides, the pentose sugar is also present in the cyclic rings called as the furanose. So the sugar present in the nucleic acids is in the cyclic form called as furanose, okay, which is a closed 5-membered ring. Okay, got it. Another important feature of this pentose sugar is that it is in 2 forms in the nucleic acids. So the recurring deoxyribonucleotide units of the DNA contains deoxyribose sugar. Okay, so DNA contains the deoxyribose, okay, and the RNA contains the D-ribose. Okay, RNA contains D-ribose. So this is present in RNA, okay, and deoxy-D-ribose sugar is present in the, okay, in DNA. Got it. So what is the difference between the deoxy-D-ribose and the D-ribose? It is simple that the deoxy-D-ribose lacks the hydroxyl group at the carbon number 2 here. So this is the carbon number 2 of the pentose sugar. It contains the hydroxyl group called as the ribose. Okay, this is present in the RNA. So RNA contains the OH group at the carbon number 2 of the pentose ring, whereas in the DNA, instead of the hydroxyl group, DNA contains the which is a deoxyribose ribonucleotide, which contains the deoxyribose sugar. DNA lacks the hydroxyl group, lacks the OH group at carbon number 2. Instead of the OH, the ribose sugar in the DNA has the H atom only. So that's why the DNA contains the deoxyribose sugar and the RNA contains the ribose sugar. So both the sugars in the DNA and RNA are in their D form, So D form means that it's the configuration of these sugars based on the spatial arrangement of OH groups on the last chiral carbon. Okay, so it is not the dextrorotatory and L rotatory. So the D in the D-ribose and the deoxy D-ribose is the configuration of these sugars based on the Fisher's projection. So conclusion is that RNA has a OH group at the carbon number 2, okay, and the DNA contains the deoxy D-ribose because it lacks the OH group. It lacks OH group. Instead of OH group, it has a H atom at the carbon number 2. So this slight change in the OH and the H groups in the ribose and the deoxyribose has the greater impact on the stability and the reactivity on these two different types of nucleic acids. Okay, so DNA has the deoxyribose sugar, whereas the RNA has the D-ribose sugar. So this slight change has the greater impact on the stability of these two molecules, and because of this change, these two molecules react differently in under the alkaline conditions. RNA is what? RNA is less stable than the DNA. So what is the reason for the less stability is that since RNA has the 2 OH group at the carbon number 2, okay, this hydroxyl group is nucleophile, okay, it's a nucleophilic. It is a nucleophilic, means it loves the nucleus by donating the electrons to form the chemical bond. So this OH group is always ready to donate the electrons. So that's why it is chemically highly reactive. So under the alkaline conditions, the hydroxyl group of the RNA acts as a nucleophile, means that it attacks the nearby phosphate group. So OH attacks the nearby phosphate group. So under alkaline conditions, the nucleophile attacks the phosphate group, and this attack leads to the breakdown of the phosphodiester bond. Because of this nucleophilic attack on the phosphate group and the breakdown of the phosphodiester bond, this leads to the formation of 2,3 cyclic monophosphates. Okay, and these cyclic monophosphates quickly degrade into the mixture of 2 and 3 nucleoside monophosphates. Got it? So which makes this RNA molecule unstable, okay, because it is easily degraded due to the presence of the nucleophilic OH group at the carbon number 2. On the other hand, DNA lacks the 2-hydroxyl group, okay. Instead of the 2-hydroxyl group, DNA has the H only. DNA has H group only. So therefore, the internal attack cannot happen in the DNA, and DNA remains chemically stable under the basic conditions. And this stability is crucial for DNA's role in the long-term storage of the genetic information. Now, coming to the final component of the nucleotides, which is the phosphate. Okay, PO4, PO3. Okay, negative. Oh, got it. So this is the last final group which is present in the nucleotides. Phosphate, it is a negatively charged chemical group. Okay, negatively charged chemical group. The phosphate is attached to the carbon 5 of the sugar. So here, this is the sugar. Okay, for example, here the phosphate is attached at the carbon number 5 of the sugar, and this phosphate can be present as mono, di or triphosphate. So if a single phosphate group is attached to the nucleotide, it's simply called as if the nucleotide, for example, is the adenine and the single phosphate group is attached, it's simply called as AMP, right? Because it has the single phosphate, so adenosine monophosphate. If 3 phosphates are attached, okay, then it's called as the ATP, because three phosphates are attached, okay, adenosine triphosphate. So remember, the phosphate group is attached at the carbon number 5 of the sugar, and it can be mono, di or triphosphate. And this phosphate group and the sugar molecule, okay, phosphate group and the sugar molecule, they together form the sugar phosphate backbone of the nucleotides. So the backbone of the nucleotides is made up of sugar plus phosphate, okay. As I mentioned that the nucleic acids are the long chains of nucleotides, and the nucleotides of both DNA and RNA are covalently linked through a phosphate group, okay, through a phosphate bridge or through a phosphate groups, in which the 5-phosphate group of one nucleotide is joined to the 3-hydroxyl group of the next nucleotide, creating the phosphodiester bond. So phosphodiester bond is a covalent bond, okay. Phosphodiester bond is a covalent bond that links the individual nucleotide units together in the nucleic acids. So phosphodiester bond links the nucleotides together in the nucleic acids, and this phosphodiester bond is formed between the 5-phosphate group of one nucleotide and the 3-hydroxyl group of the next nucleotide. So this backbone, sugar and the phosphate, in which the phosphate group links the two individual nucleotides together by a covalent bond known as the phosphodiester bond. This phosphodiester bond of the nucleotides gives the nucleic acids the directionality. So this creates the directional backbone of the nucleic acids in 5' to 3'. Okay, so this is because of this phosphodiester bond which links two nucleotides together in a way that the 3-hydroxyl group of one nucleotide is linked to the 5-hydroxyl to the phosphate present at the 5 carbon of the sugar, and this results in the directionality of the nucleic acids in 5' to 3'. So by this we are concluding today's lecture by remembering these important points is that the DNA contains the deoxyribose, RNA contains the ribose sugar, and the bases in the DNA are adenosine, thymine, guanine and cytosine, whereas in the RNA uracil is present instead of the thymine. Okay. Another important feature is that the DNA is usually double-stranded and RNA is usually single-stranded. DNA is chemically stable because of the absence of the OH group at carbon number 2, whereas the RNA is less stable because of the presence of the hydroxyl group at the carbon number 2, which acts as a nucleophile. So with this, we are concluding this lecture. Thank you for watching.